Pessoal, boa tarde a todos. A gente vai receber hoje aqui a palestra do professor Slava. O professor Slava, ele vai dar a palestra Machine Learning for Marine Sciences. Ele é PhD em Estatística pela Universidade do Estado de Orenburg, na Rússia. Professor e pesquisador associado do Center for Environmental Science da Universidade de Maryland, Chesapeake Biological Laboratory. E as áreas de pesquisa dele, as áreas de interesse dele séries temporais, a estatística aplicada, estatística inferencial não paramétrica, machine learning e análise de redes. Professor, thank you so much for being here. The microphone is all yours.
Thank you very much. Very nice introduction. Hi everyone. Bom tardes. Lo siento, yo no hablo portugués. Hi, my name is Slava. I'm not used to when people call me professor, so very official for me. Thank you very much for your welcoming environment. This is my almost third week in Brazil. I very enjoyed the country and very enjoyed people. Very nice students, very enthusiastic, and I will tell you a little bit more about why I'm here and what I'm doing. So if we can switch to Google Chrome. Yes, thank you. This is where I'm coming from, University. It's not the touch. No, University of Maryland Center for Environmental Science. It's a research institution in Maryland. It was founded in 1925. It's a public research laboratory. Chesapeake Biological Laboratory was founded to conduct research on Chesapeake Bay and target the water pollution mainly and water quality in Chesapeake Bay. And now it's a part of the University of Maryland system. And if we can scroll down. Okay. So this is the map of Maryland, very weird shape, the state, and it has several laboratories. I'm in this one called Chesapeake Biological Laboratory. It's mostly marine research on the water. Here we have Baltimore. It's a big city in Maryland, and in downtown they have a Institute of Marine Environmental Technology. Here in the west, there is an Appalachian laboratory. They do ecological research for avian species, for bats, also for global problems and for other terrestrial habitats. And here is an interesting, like across us, just what they say, 5 miles across the bay, which is about 8 kilometers. Another laboratory called Horn Point, which also does research related to oceanography, ocean science, ecology, and oyster restoration. They actually have right at their backyard oyster hatchery where they produce oysters, try to restore the habitat, and they basically plant them in the bay. Now let me switch to the slides, please. No. Sorry. And how can I switch slides? Just here? Okay.
I think you can, it's touch screen there.
Really? Oh, right. Thank you. Okay, so topic of my today's presentation is machine learning for ocean science or marine science. I compiled just several presentations, but several projects. But first of all, why I'm here? I'm here because I was awarded travel funding from the Scientific Committee on Ocean Research. Is an international organization which supports collaboration, supports working groups, research projects, infrastructure, and capacity building. So I believe I'm in a capacity building, trying to spread the knowledge or teaching in different countries. So it's a fun organization. You can check their website. And as part of this program, I have taught short course from Maringa on machine learning with students from different programs. And we had an application for students to analyze a data set coming from Chesapeake Bay. So this is, again, where the bay is located. It's very famous in the United States because it's the biggest bay in the United States. And also because there are many people living around the bay. You see the blown up map here. It covers or touches the states of Maryland, Delaware, and Virginia. And there are many people living on the coast. And also, if we consider the whole watershed, like where the water comes from, then we will also touch Pennsylvania and other go way north. The bay itself was a river before, so like many years ago, it was a Susquehanna River. And actually all the rivers have, mostly all the rivers have funny names from the names of the tribes who live there. For example, our lab is located at Patuxent River, where Patuxent River enters the bay. So I work on statistics. I'm a statistician by training. Part of my work in the lab is to collaborate with other professors on research projects, and also I teach statistics to graduate students. We are a graduate center. We have only master's and PhD degrees in environmental and marine science. And today I just wanted to present basically 3 random applications of machine learning that I have developed in aquaculture, climatic connection, and species identification. All they use machine learning at some point. So first project I'd like to present was on aquaculture. Again, the data come from the Horn Point Laboratory Oyster Hatchery. It's a public hatchery, so they were willing to share their data compared to private hatcheries who might be not willing or not conducting so much of the data intensive monitoring of their production. And the problem with the hatcheries is that they keep growing oysters. They again use oysters maybe for restoration, and they can also sell oysters for aquaculture. So to produce big oysters for restaurants, for example. And they have experienced problems with production. Sometimes when they grow oysters, they become covered with bacteria or algae, and they don't survive. What a hatchery has been doing mostly is to get rid of the bad batch, flush the tanks, clean the tanks, scrub them, and refill the tanks with water again from the environment and try to continue the production, start a new batch. There are several problems because the bad water, presumably, which was in the tanks, can still remain in the environment where they dumped it, and it can affect other oysters, and or they can actually draw part of this water again to the hatchery. And ultimately, there are no clear reasons, or there are no reasons known why the oysters die in the hatchery. So they shared the data with us to analyze. There were several response variables, so-called response variable, like what we could focus on. One was yield, is the percentage of total eyed eggs versus total eggs of oysters they put in a tank. So the higher the yield, the better. Another was the production rate. So it's how quickly the oysters grow to have eyes. I think that's when they can put the oysters to settle. So like small oysters, they settle on hard substrate, which is usually dead oyster shells. So they put them in those containers filled with the oyster shells, and then they can sell them. So at that stage. So the faster you produce something, the better. So we define the first quadrant here, number one. Mathematicians, please forgive me. In number and the quadrants, I tried, but I couldn't stand the pressure that the first should be this. So the first quadrant here is the best one, the green. So we can produce a lot and fast. Also, we can produce a lot and slower, number two. Then third worst is produce a little bit and slower, and then produce a little bit and produce a little bit and fast, and produce a little bit and very slow, the last one. So in the fourth quadrant, we lose a lot of time and don't get much in return. And also this complete die-off events, they termed crashes. Like when there is a crash, it means like there is almost zero oysters survived. So we also looked at the percent of cases. It's like a tail of a cumulative distribution here shown on the left corner. So you see that in about 15%, we don't get any yield at all. So about 15% of all the cases were crashes. So then we decided, okay, what if we look not at the average survival, but also at the so-called the lower quantiles of the observations. The data we used to analyze it represented different production stages, from broodstock collection, conditioning it in the tanks, then spawning, then producing the larva culture, and we also collected environmental data. Environmental data came from an ocean buoy from federal monitoring program. And from there particular, we could get oxygen concentration, turbidity, salinity, chlorophyll concentration, and temperature. In total, we had about from 144 to 500 observations of batches. Why the number ranges? It because depends on how many variables we used. We didn't have information for all the variables all the time. So there was a problem of missing data. Every time we decided to remove a variable from analysis, we re-evaluated the sample size and said, okay, if we don't need this variable, can we increase the sample size then? So that's why the range of the sample sizes. And first, I'd like to show you one a very interesting, I think, and quite simple method called from machine learning called classification and regression trees, CART. This method is looking for a way to split the data into most distinct subsets. And they should be distinct based on the response variable. So for example, here, we are trying to classify into which quadrant the observation will fall down if we know just the predictors. Say, if we know the temperature of water, if we know where was the larva culture for oysters taken, can we predict, can we use this information to split the data? The method is working like this. It starts with 100% of observation with all the sample, and then looks for all the observed values of each variable, trying to separate the data as much as possible. So the first split here when was picked by year the broodstock was collected. And I think it's hidden here. I think it was 2008. So if it's below 2008, we go to the left. If it's above, we go to the right. Then we can continue search again over all variables. Next was the temperature, the environmental condition. So if temperature was above 11 degrees, we go to the left. Otherwise, we go to the right. So the algorithm for you, you don't have to do much. You just specify the inputs, the variables you are trying to consider, and algorithm picks up the best splits. At the end, we have so-called terminal nodes. Here they're shown with colors. And we also see the dominant proportion in each terminal node. For example, if we went following all the splits to the very left, to the first node, we see that it contains about 8% of the data of our data set. And in this terminal node, the observed proportions of each quadrant, so there are 4, were 55, 9, 27, and 9%. So the dominant one is 55, the first quadrant. So it will be our prediction. That's why it's colored green. This is our prediction. So if every time we will see a combination of variables that corresponds to this path to the very left, we would assume that likely it will be our very good quadrant of outcomes. And for example, if the year the broodstock was collected was above 2008, after 2008, it's likely like that this observation will end up in the very bad quadrant, which was the dominant, in this case, 79% of all observations of the later years were in this quadrant. So this is, again, the algorithm. It's an exhaustive search of all combinations to split the data very well. There are some problems, of course, with this method, because you can split the data up to only one observation per terminal node, right? In this case, you will try to pick every little difference in the observation between the observations. But maybe these differences are not so important overall. Maybe if you sample a different hatchery, the tree will be very different. So there is a problem of so-called overfitting the data with this method. So you can fit all little differences which would not be true or which will not be explanatory on a slightly different sample. That's why the algorithm also applies automatic pruning to this decision tree. And the terminal nodes are not very small. So here we see like the percent of observation ranges from 6 to 22%, or 27 actually, 27%. So we are trying to balance goodness of fit with overfitting. It's a typical problem in regression. For example, if you're selecting a regression model, you might be using, say, Akaike information criterion to select the model and to balance the goodness of fit of the model and number of parameters or complexity of the model. So here the same problem is solved, which sometimes it compromises the goodness of fit. So one of the solutions is to use many trees where each tree is overfit to the data. So we allow each tree to be very deep or very big. We will overfit and fit the sample data very well. Maybe we will not be able to generalize well to other population, but by using many trees, like in a forest, we will be able to generalize and have more stable results. That's the idea. And that's why we used random forest, which one of my favorite machine learning methods. So if you're just starting to apply machine learning, start here with random forest. There are several methods and or types of random forest. If you are trying to classify like quadrants, it will be random forest for classification. If you are predicting a numeric response, it will be random forest regression. Or if you are trying to predict numeric response, but not at the mean, like usually when we fit regression, we are fitting the mean, right? Average response, how average changes if the factor changes. But you can look at the quantiles. So quantile regression, you can apply quantile random forest. So for the first two, numeric and categorical, almost the same packages are used. For quantiles, there is no much correspondence of packages. So I had to like switch to different and even write my own combinations to say select variables in quantile random forest. So this is just example of packages. In this approach, I use the following pipeline or the process to analyze. So first we can tune the random forest, like in say in statistical model specification, you can choose different polynomials or different specification of the model. In random forest, we can select several parameters which are not directly estimated from the data, which we set manually. They are called hyperparameters, like hyperbolic or hyperparameters. And this is number of variables we try randomly at each tree split and the terminal node size. The good thing is that in many cases, the default values work well. So you can allow random forest to grow deep. So you can set very small minimal node size. And mtry is the number of variables we try. Also, the defaults work well. Usually it's about 1/3 of total number of observations. So if we have 20 or like 18 number of variables. So if we have 18 or 20 variables, we will be likely selecting the best split, not among all of them, because it will be very computationally intensive when we grow also many trees, but only about 6 variables selected randomly. If we go deeper, there is a reason behind that, why we also select a subset of variables. It's not just for efficiency, but also to make the trees more different from each other. So we have different opinions, right? It's like making sure that we have different opinions, that we can generalize them better compared to having very similar trees. What's the point of having very similar trees if they all already agree, right? So we can tune these parameters additionally using cross-validation. So we can, what is cross-validation? If you are again new to machine learning, is a way to split the data into folds. Here I'm splitting 10 parts or folds the data, and each time you use 9 of them to fit the model and try to forecast on the 10th. And for the 10th one, which you didn't use to fit or select the model, you can compare observed values with predicted and calculate the errors. And then you can repeat it for another combination and another combination of folds, so you can estimate the errors and repeat it again for different set of parameters. So you don't estimate the parameters directly, but you do it by some kind of grid search. You search over different combinations and select the best one. Then there are additional algorithms to select variables in machine learning. Many of them include additional permutations. So you can randomly resample the data and try to see if, for example, if you resample one variable, will your results change? That's the basic of many algorithms. If they change, it means that variable was important, right? If the results don't change, that variable was not important for predicting the response. Then after variable selection, we drop some variable from the model. Then I would increase the sample size if possible, and then repeat the random forest tuning, and then finalize the model. That was my pipeline. As the result, you can see which variables are removed from those 20, but here maybe just look at the accuracy. So for numeric response, the R-squared, or the percentage of explained variability, was 54%. For predicting the quadrant, the accuracy was 60%, which I consider was good, actually, because if we come back to the original plot, there were many cases on the edge. No, sorry. Oh, it's okay. So there were many cases on the edge, and there were four classes, right? So it's not just binary prediction, 1 or 2, 1 or 2. So accuracy 0.6 is, I think, acceptable for this kind of data. And then I also show the R1, which is the way we can quantify the errors for predicting quantiles. So this is like a absolute value of the error. What fun about random forest is that then you can output the way sort of the variables are modeled in this algorithm. This is called partial dependence plots. And what they do is they vary the values of one variable while the rest of the variables remain fixed. And we have this partial dependence or partial residuals results like sometimes we have for additive models in statistics. So, for example, what we can see from these ones is that I think there was interesting results For salinity, so for salinity, the last plot, we have like a sweet spot, higher yield when salinity is about 10. Then we can see that also the yield drops later in the year when the week of the year is increasing. And also there is some optimal region for, say, temperature. Some of the variables like percentile of survival to dehinge, so this is a developmental stage in oysters, cannot be controlled. They have very strong relationship, but we cannot say to oysters, oh, please survive, right? We can calculate the percentage, but to control it, we need to run a separate analysis. Another way to output, here we output predicted quadrants, and these are 2D plots, so you can also see the combination of variables. It's a little bit more noisy graph, but for example, what we can see here is that if percent survival is high, then, well, possibly doesn't matter of the salinity, we can go to the first quadrant, which was good. Unless the salinity is really high, like here we have a blob of the quadrant number 4. And I personally like the smile here, the emoji. So, of course, you wouldn't pay attention to this pattern like the emoji one, because machine learning, the essence is to find patterns in big data sets. And our data set was probably biggest in the United States for this kind of problem, but not as big as typical machine problem look like when you are looking for patterns, say, in Facebook communications and so on. These were the plots for quantiles. So you could see that, for example, the lower quartile for oyster yields, how it changed with salinity, and the upper quartile. So, and the same with turbidity of the water and oxygen concentrations. So these are some results. I'm sorry, it's too many plots for the same, but if I go to conclusions, we tried to show importance for the hatcheries to collect and share the information. We tried to create a network of hatcheries so they can send the data for analysis and receive some feedback, and also if they can use, and if we can use their data to further develop models, that would be good. So we are trying to develop these data-driven methods to identify important factors, because even ranking them by importance is useful. So we can further run some experiments and really understand why hatchery crashes occur. In the future work, we also try to develop predictive models, to tell in advance if the crash is likely to happen, because some of the variables I showed here, they are known, say, exactly when you already see the crashes happening. The idea is, what if you know that it's likely, maybe you can dump the water early and don't waste time and money on heating or cooling it, etc. And of course, increase sample sizes. Oh, people usually like. Any questions? Do you work with aquaculture here? Yes?
Yes, we do. We actually have a course, aquaculture engineering. So there are some students of aquaculture too.
Nice.
I have a question. I don't know if it's better to save the questions for the end or now, but how intensive, computationally speaking, are those random forest methods? They are running in a computer, regular laptop, or you need a special computer to do?
That's a good question. For small problems like this one, our packages are very efficient. It seems like I I don't know how to operate it. So in thank you. Can we go to the table? So there are a couple of popular packages. One is called Random Forest, and I think it's written in R, and it runs fast, so it wouldn't take more than a minute. But another package, Ranger, is written in C++. The whole syntax is still in R, but the operations are done in C++, and it's much faster. You probably wouldn't notice the difference on this data set of like 500 observations, but for bigger problems, it makes a huge difference.
It depends, of course, the quantity, the volume of data you are analyzing. You said your data set is not that large, so it runs faster with a small data set. It's an interactive method.
Right, because you would need to do additional permutations if you want to do like variable selection, or if you need to do fine tuning to select like some of the hyperparameters of random forest. More questions? Okay, so the next project I wanted to briefly touch about time series analysis, and I think I have only a couple of slides here for this one. This was developed with my colleague Halle Kilborn. Sorry, Halle Kilborn is a paleoclimatologist. So one of her passions is to drill corals, ancient corals, and recover the temperature time series from chemical concentrations. And she also worked on compiling big data sets from multiple researchers, so to have international big data repository from all different people who did the same work. So the so-called paleoclimate data proxies are available for download. And what we wanted to look at was the climate connections between North Atlantic Oscillation Climate Index and the temperature series in different locations of the ocean in Atlantic. As you know, with time series, one of the first steps was to remove trends before we tried to correlate. And basically, we removed long-term trends with about 200-year length of lowest filter. Then we wanted to correlate this time series. And you know, when there is a still remaining autocorrelation, you don't have the same sample size, or you don't have the same confidence as if you have independent observations. So we wanted to account for remaining autocorrelation and wrote a function to create confidence bands using Bootstrap for correlating time series. Next, the correlation was already some progress in that field, like to see which time series correlate with each other. But also, when we deal with these climate proxies, we often don't know exactly to what year the observation corresponds to, because it's not a direct observation, right? Where it's like tree rings on a tree. So we don't know exactly what was that year. There are some methods in paleoclimatology to so-called date the record, so to assign the date. But then there could be differences between corals or between different researchers. So to avoid that, or to account for that, we applied the method of dynamic time warping to match the time series. This method is from machine learning, particularly from voice recognition, because sometimes you can say the word elephant. You can say it quickly, you can say it slowly. It's the same word, but if you try to find Euclidean distances between two recordings, first, you don't have enough overlap sometimes, right? If you said the word in a long way, it will not cover the short, it will cover like the short version, plus you'll have something remaining. And if you find the Euclidean distances, you will find the big differences between these two recordings. So the method of DTW tries to find the shortest distances by kind of allowing the time axis to be flexible, so it can stretch and or squeeze in time the recordings. In this case, we can match two climate proxies in time. So the two lines here are showing different climate proxies from different corals, and the unseen lines, sorry, they're very faint, correspond to matchings. So the idea, for example, that this minimum can be matched to that minimum, even they don't happen in the same time, but approximately in the same time. So the algorithm picks these matches. You can put different restrictions on that, like how far you can select the matches. And then you can calculate these Euclidean distances, but not in exact matches of time. So this will be the distances between recordings. When we have the distances between recordings, we can cluster, right? So we have the distance matrix, and then we applied some clustering technique, which then whichever you like. It could be, in this case, I think we applied hierarchical clustering, selected the best method. You can choose how you join the clusters. And then you can identify the cluster and then try to apply analysis on that cluster in particular. What we applied, we developed a test for long-term grandeur causality. It's a statistical method, so I don't want to spend too much time on that, but the idea is that we try to find which time series is predictive of another one. So we have a cluster of water temperatures. Can we say that these water temperatures in this cluster are predictive of NAO, North Atlantic Oscillation, say 30 years ahead or so? So while we say long-term, we remove the short-term lags. That's the modification of the classic grandeur causality method. So we use restricted models to remove certain lags and then test the hypothesis. So in this project, we apply this dynamic time warping, the machine learning technique, and continued with statistics. Some of the findings, so I applied it to, without clustering, we were able to show that NAO affects with 3-year lag tilefish landings in Atlantic. Landings is like how much fish you brought right to the shore. And 3-year lag is possibly can be explained by the fish was affected when it was young, before it showed up in the landings, right? So the small fish was affected by some temperature changes. Then later it was visible in the amount of catches. And in the work which is now in progress, we couldn't see the statistical effects of statistical causality among the observational like proxy data, but after we clustered, we got some statistical significance in the results. So we think it's a promising method to aggregate information from disparate researchers and find signal in noisy data, because we can aggregate information more efficiently with dealing with time series with uncertainty in the time itself. So this is my second thing. So like the next steps, we want to develop a causality network for Chesapeake Bay. So analyze how things spread in time by correlating the time series in the bay. And the next one, which I think one of the funnier one, is species identification. Helen Bailey is our researcher in marine mammal acoustics. One of the things she does is put in hydrophones in the water to record whatever and whichever. There are different types. Some record certain frequencies. Some are also then aggregating information. If you are aggregating and you don't record the whole sound signal, then you can record for longer periods, so months, compared to just several days, right? Because you don't need so much power, battery to record. Here's a boat, but actually most of the data are coming from like static recorders when the recorder is at the bottom or near the bottom of the ocean. We are currently doing it for monitoring before the construction starts for wind turbines, because when they build the wind turbines offshore, they need to comply with the Protected Species Acts, like make sure that there are no endangered species in the area. So the monitoring helps to analyze occurrence and reoccurrence of whales, dolphins, and other acoustic generating species. And also we have Helen has the online application and cell phone application called Dolphin Watch, where users can upload pictures of dolphins and report seeing a dolphin. And it also helps to analyze the patterns of dolphin occurrence in the area. But what we've been doing is trying to automate processes of analyzing the acoustic recordings. Here's a spectrogram of passive acoustic monitoring with the frames showing manually identified dolphin whistles. It's a very time-consuming process, and basically a trained acoustician should listen to recording. Not me, but some trained person should listen to recording and say, like, this is a whale or this is a dolphin, and mark the recording. And my work would be to take this labeled data. So when we have the signal and we have the information about what we are looking for, so like this is a whistle. So we need to automate and design the algorithm which will do it automatically. We used R for that. This is if we are developing an algorithm, we will just use some standard chunks of recording. So for example, if we use 1 second time chunks, this is a spectrogram for each 1 second. And we have hundreds of them or thousands of this kind of spectrograms. These are manually selected spectrograms which actually have whistles. If there is no whistle, if there is just noise, you'll just see the gradient from blue to yellow. And then we are trying to classify them. Imagine like classifying if there is dog or cat on an image. So these all spectrograms should be classified as presence or absent versus the absence of a whistle. For that, we are using technique called convolutional neural networks. This technique processes the input, the image, through a series of mathematical modifications. They are called layers. Each layer contains so-called activation function, which is nonlinear function which transform the data. If you are familiar with generalized linear models, it is like a link function. So it transforms the data non-linearly and in many different ways, and then aggregates this information. If you look, some of the boxes become narrower, is because, say, image was 100 by 100 pixels, then we find averages or maximum within a 4 by 4 pixel range. So we're aggregating. We're trying to create these bottlenecks to learn the general features more efficiently. It's the same as I told the students in the class. If you are trying to describe me your city or your country in one word, like try to be as more efficient as you can to describe something. And then the second part of the network will try to classify it. So you'll reduce the information and then we'll classify it into, well, this is a stolen image. We are classifying car tracks, but in my analysis, we would classify dolphin present or absent. We applied this data. We had nice recordings from Baltimore Aquarium. They led the Helens group to put hydrophones and record dolphins. But unfortunately, there were not. It was not a big data set. So here we have results for training models on Baltimore Aquarium data, and then we use some existing model called Dolphin Spot, developed by Scottish researchers. So the results are promising because actually this analysis was done by my summer student, bachelor student who had an internship at our lab. So this is her results. The results are promising because we had very few observations in the Baltimore Aquarium compared to the Scottish wild populations. And now we are working on basically retraining our models on our bigger data set because the Scottish model was not as good as we expected on our data of on our Maryland data. And so that's one of the reasons, like captive dolphins were not as good to train our models. And also from the results, we saw that ResNet 50, so the model with 50 layers, performed better than a simpler model. So we will probably stick to a bigger model for image classification. And next, we want to classify species also to distinguish between, say, common dolphin and bottlenose dolphins, which are common in the Maryland area. And we will also try to identify individual animals because dolphins have unique signature whistles, which remain almost unchanged during their life. So they basically speak their name when they communicate with other dolphins. For example, I say, "Hi, I'm Slava." So, and you can identify the signature whistle in the dolphin. There are some problems when dolphins are in a big group, but we will work on that as well. So the idea is that when we know signature whistle, right, we can help ecologists, biologists to identify the group size. How many dolphins are swimming, right? Or maybe we can also identify the migration patterns if we have an array of hydrophones and if we, say, share information with, for example, other locations like Scotland. What if we can spy on dolphins? So these are my extracts. I hope I didn't bore you too much. And thank you for your attention. How can I know what the best algorithm to use based on my database? Which the best language to use in machine learning? Oh, okay. The first question, the best algorithm. For that, I would to select that. Of course, literature search would help to start with something that people are already using as a baseline, and then you can try to improve it. Use a different algorithm. You are probably just limited by your imagination at this point. But to compare them, typically people use cross-validation. Again, splitting the data into folds and trying to predict with both or several algorithms and compare the errors. The lower, the better. Which best language? I'm using R, but in some cases, R serves for me as just interface. For example, the later models like convolutional neural networks are in Python. So R calls for me Python libraries to fit the model, and then the results are again presented in R. I don't use Python directly. So if you just want to use machine learning and like any language, then maybe Python. But if you are R user, you can stick to R. Stick with R. And thank you for writing in English.
In our third application, thanks. In your third application, you used the periodogram. What kind of periodogram it was? Because it seems it's not just in time. It seems it has a spatial time periodogram.
Thank you. That was a fast Fourier transform. So we used a 1 second interval for the sound, and then the frequency range was divided into. I don't remember exactly. Yes. So then within that, within that 1 second, we also had, for example, 512 steps. So we divided 1 second into the 512 steps. So that was the time dimension. And then for the frequencies are on the vertical dimension, and the color represented the power of the spectrum. So each second was further divided into substeps. It was possible because the sampling rate was high for the recordings. So the sampling rate is for recording the sound. It's a fascinating topic, but like because we don't record the sound continuously, we have actual time, like discrete time for like which is called sampling rate, like how many samples you take per second. So our sampling rate was about 48,000, I think, per second. So it was possible.
Thanks for. And maybe in future, I don't know if you have it, but if we use Wavelet, maybe we can get more information from this because of naturally Wavelet can see that in the best way that will be transformed. But I don't know, maybe because probably you get when the dolphins say hi and the name, maybe they are saying at the same time, and maybe we could separate one from each other. I don't know.
I agree. Actually, it's a great idea. I know wavelets will be great for this spectrogram representation. As you saw, like our pictures are not as bright, not as contrasty compared to the big picture, like the original one I showed, which was from, I think, from commercial software, where they listen to like manually and label for us. So we tried to manually like mix channels because our spectrograms are recorded in red, green, blue. So we tried manually improve the contrasts of the images to classify better. But maybe we should just change the mathematics instead of manual manipulations. Do you use machine learning only for acoustic identification and species occurrence? My PhD is exactly about dolphin acoustics, but in my case, I would like to identify and compare the structure of the whistles, its contours, almost like the study with signature whistles. Do you have any tips regarding codes? Okay. I frankly don't have tips. I'm familiar with some papers which try to contour the whistles, so they draw a contour around the signature whistles, and they create new variables to characterize it or to describe this contour. I think there is a method on using this approach called banter, but it's actually based on random forest. So the idea is that when you manually identifying these characteristic features of the contour, you are manually creating features or variables for your research. And when you have several characteristic features, like say 5, which describe the contour shape, you can just use this variable in almost any method, such as random forest. And one of the applications is called banter by I think there are NOAA researchers at the United States, so they use random forest. But we don't create features manually. We let the neural network to do it for us, to learn this short representation of the image. So I don't have recommendations about coding the contours because I try to avoid it. Thank you.